Hello, everyone. As a postdoctoral researcher at the Princess Margaret Cancer Center, it is a real privilege for me to share with you my work on breast cancer prevention. If we look at the year 2024, of the new cases of cancer diagnosed among women, a quarter were of breast cancer. That's 30,500 women. Now, improvements in screening measures mean that we're getting more timely diagnoses and more improved treatment of patients. But not all cancer cases are equal. What about stage 0 cancer or pre-breast cancer? This is also known as ductal carcinoma in situ or DCIS. DCIS is characterized by cancerous cells that are contained within breast ducts. DCIS may progress to a more invasive or more serious breast cancer, which is why physicians typically prescribe mastectomies, life-altering surgeries to these patients. And yet, it is hard to assess when a DCIS case is more serious. And this is especially significant as patients risk a high risk of recurrence years after preventative surgery. This obviously causes a lot of psychological distress, especially given that clinical parameters like breast density are imperfect. So I propose to look beyond clinical parameters and to look at molecular subcellular differences between DCIS cases that are more severe and others, especially looking at metabolic differences. Now, whether healthy or cancerous, cells use metabolism to acquire nutrients from the environment, so things like oxygen and glucose, from, say, blood, to produce energy and other building blocks for proliferation, and employ enzymes, which are protein molecules encoded by genes within the genome, to make this conversion from input metabolite to energy and so on. Now, in contrast to healthy cells, proliferating cancer cells are characterized by abnormal, rewired metabolism, where they utilize a lot more glucose to ultimately get the same energy yield. This is paradoxical, but makes sense when you realize that human metabolism is actually highly complex, where the products of energy metabolism, in effect, feed into other metabolic pathways. And so, in effect, what you see is that energy metabolism is balanced off with other metabolic pathways, say, that produce the building blocks required for the uncontrolled proliferation that characterizes cancer. How do we decipher this? I use computational techniques to understand this majestic hairball that's human metabolism and to understand what's happening at the subcellular level, particularly metabolic modeling. And I am using data from the Translational Breast Cancer Research Consortium concerning 158 DCIS patients. These patients underwent surgery after diagnosis and were followed over a period of at least 5 years. And over the follow-up period, 95 had no recurrence, whereas 63 experienced DCIS recurrence within 5 years of diagnosis. In particular, I am using gene expression data collected from the breast tissue that's dissected during surgery. Now, when a gene from the genome is expressed, it serves as a readout of how much the corresponding enzyme is actually active in the cell, and thus what metabolic functions are being used by the cell. So I identified 529 genes differentially expressed in the DCIS recurrent cohort, mapped this onto this majestic network, and through modeling, identified that fatty acid synthesis is upregulated in the DCIS recurrent cohort. And this aligns with previous findings of increased fatty acid synthesis in breast cancer. Importantly, this serves as a marker of potentially DCIS recurrence, which requires further work. Now, I will expand on this work to look at a bigger cohort and identify ancestry-specific signatures, so that we can better predict DCIS for different individuals of different ancestries. To end, Dr. Mukherjee called cancer the emperor of all maladies, the king of terrors. I hope that with my work, DCIS patients have a better chance of fighting recurrence and a better quality of life. Thank you all.